Okay, so everybody please turn into the amino acids PowerPoints. And we said that the amino acids are the building blocks of the proteins. And we said that they all have the same three substituents. If you're in the back, you can look at that monitor there. And if you are here, where is this? There we go. Okay. So nutritionists, those are the nutritionists. They have classified amino acids into essential and non-essential amino acids. Essential amino acids are the amino acids that we need to take from diets. Your body cannot make on its own. And we rely on our diet in order to make them, while the non-essential amino acids, your body is usually able to make them unless you are sick. If you're sick, even the non-essential amino acids you cannot make. You absolutely do not need to memorize which amino acids are essential, which are non-essential, but you can see that the vast majority of the amino acids are non-essential. So what are the elements of the amino acid? If you see here, we see that there's the central alpha carbon. Remember, I drew that. The alpha carbon is the central carbon, and then it has four separate directions: north, south, east, and west. So all the amino acids share the same three substituents, which are the hydrogen, the amino terminal, the carboxylic terminal. Only the R substituent is what makes the amino acid unique. And that R substituent, when combined with other amino acids, is what's going to determine the primary structure and the folding. Why do some amino acids have a alpha helix? Why is another amino acid has a beta pleated sheet? It's because of the R substituent that is part of all these primary sequences. So you need to know your alpha carbon. This arrow here points to the alpha carbon, and the alpha carbon is usually for most of the amino acids, except one, is chiral, C-H-I-R-A-L. What's chiral? Chiral means that it is bound to four different substituents. Like here, our substituent is different from the amino, is different from the carboxylic, is different from the hydrogen. Now, this is known as the alpha carboxyl terminal. That's the alpha carboxyl terminal. That's the alpha amine. We call it the alpha because it's attached to the alpha carbon. And this is the R group. There you go. Look at cysteine here. Where is your alpha carbon? It would absolutely not be this one because that's the alpha carboxyl group. It wouldn't be this one. That's the alpha amino group. And this is the hydrogen. Hydrogen is always invisible. It cannot be determined, but it cannot be depicted in this type of particular model, this depiction. But here, sulfur containing, it shows you that this is your alpha carbon. That's the alpha carbon. That's the alpha carbon. That's the alpha carboxyl. That's the alpha amine. And this is the R substituent. Look at the R substituent of this particular amino acid. It has a ring, an aromatic ring. Aroma, like they use it all the time for the perfume. That's the aromatic ring. And this is the alpha carbon. This is the alpha, not this one. You need to know which one is the alpha carbon. The alpha carbon is this one because it is bound to four different groups. Where is the first group? That's the alpha carboxyl. That's the alpha amino. And this is the R substituent. Where is the hydrogen? It's invisible. Usually we don't make hydrogen groups. Okay, that's the alpha carbon. Now, all of the amino acids are in the L form, and they have an asymmetric carbon. And this is the categorization of the amino acids. We see that some of them are nonpolar, meaning they dissolve in lipid. They do not dissolve in water. Carboxyl, meaning that their R group has a carboxylic terminal. So in addition to having a carboxylic terminal, the R group can itself be carboxyl again. Some R groups are amine. In addition to having an amino group, they can have an amine R group. Some of them can have an aromatic ring in the R group. Some of them can have a hydroxyl group, an OH. So now we're going to talk about the ionizable groups. These ionizable meaning can undergo ionization, become a cation, positive cation, losing an electron, or a negative anion, gaining an electron. As the aspartic acid and the glutamic acid. This is a negatively charged, while the lysine, arginine, and histidine are positively charged. Then you have the tyrosine, has a negative oxygen on it, and cysteine. And this is an aromatic group. An example of that. Absolutely, you do not need to memorize which groups are aromatic. That's just for you to see that some amino groups have always the most distinctive group that define the amino acid is the R group. One amino acid is hydrophilic, one amino acid is hydrophobic. It's mainly because of the R group. You see, that's the R group. Okay. And you see that we're neutral because the oxygen is negatively charged while the amino is positively charged. So they cancel each other out. So you know the amino acid has two terminals. What are the two terminals of the amino acid? What are the two terminals of the amino acid? The two ionizable terminals. You know the amino acid has four terminals. Hydrogen, R substituent, amino, and carboxylic terminal. It's the carboxylic terminal and the amino terminal. Those are the two ionizable groups. So they cancel each other out. So the carboxylic terminal is the acidic terminal because it donates the hydrogen proton. The oxygen terminal, the carboxylic terminal, is the terminal that is able to donate a hydrogen proton, while the amino terminal is the terminal that is able to gain a hydrogen proton. It's able to gain a hydrogen proton. So that shows you that the amino acid is a buffer, because in your body, if the acidity goes up, if there is too much hydrogen, if the acidity goes up and there is too much hydrogen proton, what does that mean? I'm just gonna have the phone. I forgot the phone completely. Thank you so much. So what's the definition of an acid? Who can remind me about the definition of an acid? Ask me. What's an acid? An acid is any substance that is able to do what? Donate hydrogen proton. And a base is any substance that is able to do what?
Donate hydroxyl.
Donate hydroxyl.
And it accepts hydrogen.
Or accept hydrogen proton. Excellent, has two definitions. Okay, so if you look at the amino acid, what is an amino acid? Is it either or can it be both? Because remember, the amino acid has an amino terminal and a carboxylic terminal. The amino acid has an amino terminal and a okay, the amino acid has two terminals. That's one terminal, the carboxylic terminal, and one terminal here is the amino terminal. The amino terminal and the carboxylic terminal, which one of them is acidic? You think the carboxylic terminal is acidic or the amino terminal is acidic? Who's acidic? The carboxylic terminal from carboxylic acid. So if your body is suffering from alkalosis, your carboxylic terminal is going to donate a hydrogen proton. And if the body is suffering from acidosis, then the amino terminal will act like a base and it will reuptake all of the hydrogen protons. This shows you here that the R group, our substituent, is responsible for defining the amino acid. Okay, remember I mentioned to you that all the amino acids are chiral. Chiral means what? What does chiral mean? Chiral. I think somebody's saying it. Yeah. Excellent. It means that the alpha carbon, the carbon in the middle is called the alpha carbon. Chiral means that the alpha carbon is connected to 4 different substituents. Well, one amino acid has an R group, which is a hydrogen also, like our friend glycine. If you look at glycine, where is the alpha carbon? It wouldn't be this one, because that's the carboxylic terminal. That's the amino terminal. That's the alpha carbon, and it is now connected to a hydrogen substituent and a hydrogen R group. So when the hydrogen R group is when the R substituent is itself a hydrogen ion, then that shows you it cannot have 2 hydrogens. So it's achiral. So what's the only amino acid that is achiral? That's the glycine. See, glycine is the only achiral. One amino acid has its R group making a ring with its own amino terminal. Can you look at it here? Which amino acid has an R group that is connected to its own amino terminal? Proline. Okay, so proline has an R group that is connected, making a ring with its amino group. And once you expose yourself to all the amino acids, you will get a touch of which one is polar, which one is non-polar. But you remember, what makes an amino acid polar or non-polar or aromatic or negatively charged or positively charged? What gives it that property? The R substituent, the R group. Very good. So there you go. Those are the R groups. You see, glycine has an R group who also happens to be what? Hydrogen, which makes it chiral or achiral? Chiral. So that shows you the carboxyl terminal is negatively charged and the amino terminal is positively charged. Okay. The glycine, yes, because it has an R group who is a hydrogen. And remember, all the amino acids have to have a carboxylic terminal, amino terminal, and a hydrogen. So now it has 2 hydrogens. So it is not bounded to 4 different. Okay. This one. So it has, this is glycine. It is bounded to a carboxylic terminal, an amino terminal, an invisible hydrogen, but its R group is another hydrogen. So that makes it not bounded to 4 different. So it's not chiral, it's achiral. Aspartic acid has a negatively charged. Remember, always the carboxylic terminal is negative and the amino terminal is positive, so they cancel each other. But aspartic acid has an R group who is negative. So when those 2 cancel out, there is an overall net charge that is what? Negative on the aspartic acid. Glutamic acid is negatively charged. Look at serine, threonine, and tyrosine. Carboxylic and amino are both going to cancel out, but serine has a hydroxyl group. Is it charged? Is hydroxyl charged? So that's a neutral. Serine is neutral, threonine is neutral, and tyrosine is also neutral. Cysteine, something that is very special about cysteine is you need to know that. Remember, the same way we learned that something very special about glycine is achiral, its R group is is our substituent is hydrogen. We also learned that proline has an R group that has a ring with its amino terminal, right? See, that's asparagine and glutamine. And you see, this is histidine, lysine, and arginine. And these are all groups that after the two terminals cancel out, there's going to be an inherent positive charge. Like histidine has, remember we talked about asparagine and glutamate, both of them being negatively charged. You see here, serine and threonine are neutral, while aspartic acid and glutamic acid are negatively charged. Aspartic acid and glutamic acid are both negatively charged. But if you examine here, you see that some are positively charged, like arginine. Arginine has an inherent positive charge after both cancel out, after the carboxylic terminal and the amino terminal cancel out. There are rare amino acids. Why are they rare? Because they are encoded by a stop codon. You know, when we're performing translation, you can get to a stop codon, and the stop codon is going to cause termination of the translation sequence. Some amino acids, that stop codon makes them, which is right here, pyrrolysine and selenocysteine. But you need to remember that cysteine has a special what? Sulfur S group on it. Non-protein amino acids. These are amino acids that are not part of a protein. Just because amino acids are the building blocks of proteins, that does not mean that every single amino acid out there is making some protein. Some amino acids, they function on their own, like ascorbic acid. You know, vitamin C, that's an amino acid. Is ascorbic acid a protein? So not every amino acid is going to make a protein. Every protein is made up of many multiple amino acids, absolutely, but not every amino acid out there has the intention of becoming part of a polypeptide or a peptide. Are we clear? Okay, so if you look here at the ionization, you see that the more we add of a base, the more the pH increases. And as the pH increases, what's going to happen? You see that as the pH increases, there is an ionization. So you see that as the pH increases, the carboxylic terminal is going to lose a hydrogen proton. Then we increase the pH, then this terminal is going to lose another proton. And you increase the pH, then the NH3 is going to lose another proton. So that shows you that these groups are ionizable and they are affected by the change in the pH. The more the pH increases, the more deprotonation, the more we're going to lose the hydrogen protons. Okay. All righty. So we learned that enzymes, in order for them to operate, they need a proper temperature, they need a proper pH, and they need the ATP in order for them to operate and to do their job completely. You see here, it's the cause why some proteins need the proper pH, because they will denature. You know these protein enzymes, they are like quaternary structure or a secondary structure. If you don't put the proper temperature, if you don't put the proper pH, they will denature and they will fall apart. So that shows you at pH 2.5 or pH 3, the activity of pepsin. What is pepsin? Pepsin is an enzyme that digests proteins. And trypsin is another enzyme. Pepsin is secreted in your stomach. Your stomach is secreted by chief cells. Chief cells secrete pepsin. And parietal cells secrete hydrogen protons, the acid. So pepsin is working at very low pH. It's an acidic pH. The maximal activity, the optimal performance of pepsin is when the pH is low, while the optimal performance of trypsin is when the pH is almost near neutral. So that shows you it depends on the R group of the amino acids that make up that particular protein and their ionization. We discussed post-translational modification, which are changes and surgery that we make to the mRNA. Remember last week, we said there is extra exons and introns. Introns are going to be left behind in the nucleus, while exons are going to be exported into the ribosome to be translated. Now, if you look at the if you look into the post-translational modification, sometimes we add a hydroxyl group. Sometimes we ionize and we add a carboxylic acid group. That's all a post-translational modification. That was not part of the ribosome. The ribosome did not print out for us a protein with a hydroxyl group. No, we added that as a post-translational modification. What's another type of post-translational modification? See, hydroxyproline. You have a proline with a hydroxyl on it. Carboxyglutamate. That means you had glutamate and we added to it a carboxylic group. Carbohydrate asparagine adduct. So you add an entire carbohydrate asparagine adduct to this particular amino acid. Sometimes there are phosphorylated amino acids that you add phosphate to it, like phosphoserine, phosphothreonine, phosphotyrosine. Tyrosine is the actual amino acid. Phospho is the modification, is an auxiliary, supplementary part we need. And you know, all these modifications are important for what? Why do we have post-translational modifications? For the folding. They affect the folding. What type of group that you added? A phosphate group, a hydroxyl group? Did you add a particular adduct? Did you add a amino group? That's gonna play a big role in the folding properties of a particular protein. Okay, you need to remember that amino acids are broken down into in catabolism through the endothermic breakage of the bonds in order to liberate the energy to exit out, and that is an exergonic reaction for our catabolism. And that shows you here glucose, 6-carbon molecule, as we're going to learn in carbohydrate metabolism. We're going to break it down into pyruvate, and then pyruvate can be used as an intermediate. I need you to remember something that I said earlier in this lecture. I told you that carbohydrates can be stored as carbohydrates. Fats can be stored as lipids and fats. Who is the only one that cannot be stored in their original form? Okay, so you see here, tryptophan, tyrosine, phenylalanine, threonine, isoleucine, lysine, leucine. Are those amino acids or are those carbohydrates? So look at what the body does to them. Converts them to acetyl-CoA, then shuttle the acetyl-CoA into the Krebs cycle, and the Krebs cycle is going to shuffle and cause the metabolism, and then finally convert oxaloacetate into phosphoenol pyruvate, and then we can convert it into glucose, we can convert it into pyruvate. So you see there is a direct linkage between, you see all these amino acids, arginine, histidine, proline, threonine, isoleucine, tyrosine, phenylalanine, aspartate, all of them are shuttled in this very important cycle. What's the name of the cycle? The tricarboxylic acid cycle, or the Krebs cycle. And you see the Krebs cycle is going to take all the amino acids, do surgery to them, modify them, and then exit them as what? As acetyl-CoA and phosphoenol pyruvate. Once you have phosphoenol pyruvate, then you can go either way. You can become like glucose. Remember, they used to be amino acids. Now they have been converted into glucose or pyruvate, tomato. We can, whatever the body needs. And if you can see here, there are pathways that convert alanine, cysteine, glycine, all amino acids, convert them into pyruvate. So you see now there is a direct link between breaking down the amino acids and bringing them into the Krebs cycle. Krebs cycle. The peptide bond formation, we discussed that. That is very central. You need to know that two amino acid 1, amino acid 2, you see the R substituent is what makes them different. Amino acid 1 is different from amino acid 2. It has everything the same except the R group. So if I want to bind together, I remove water. So one oxygen and hydrogen are removed from the carboxylic terminal, while hydrogen is removed from the amino terminal. Then I make my peptide bond through dehydration, synthesis, anabolism, bond formation, endergonic. Heat has been removed, exothermic. Any questions? All right, that's the amino acid done. Let's move on now to the next one. What's going on? Any question? I'm sorry? Okay. All righty. Proteins. So let's now. Okay, everybody, please turn to the proteins. Everybody's on the proteins? Okay. So, you know, we make the peptide bonds. We discussed that. The peptide bonds are important. Why? Because these peptide bonds are able to store the energy. Amino acid 1 and amino acid 2, they were lonely, had no energy. You know, when people get married, they have energy? No. You know, the tax income. They take so much tax from me, now I want to get married. Okay, so those two amino acids, they were separated. But when they bond together, they have so much energy. They have a big account. They have income return now. They have a lot of money now. So that is anabolism. So you need to remember, we're not only going to see this with proteins, we're going to see this with carbohydrates. We're going to see this with nucleic acids. We're going to see this with lipids. So you see, we form the bonds. You need to know how this bond is formed. It's formed between the alcohol OH of the carboxylic acid. You need to know who gives what in the relationship. Because you know, in the end, water came out. You know that it's called dehydration synthesis. So we synthesize by dehydration, by the removal of water. We remove water in order to bind the two amino acids. So you need to know that these are two separate amino acids, 1 and 2. The one that has a carboxylic terminal will donate an OH, and the one that has an amino terminal will donate 1 H. Now you have O and 2 H's. What does that look like now? That is water. OH with an H, that's water. We devoid water, we remove water. What do we call that? We call that Dehydration, synthesis. We made a high energy bond now. So the alpha carboxyl of one amino acid and the alpha amino of the other amino acid. You're not going to make a bond within the amino acid itself. It has to be between two different amino acids. Okay, so that's the peptide bond. You need to be able, if you see a molecule, you should be able to look and tell which one is the alpha carbon of the first amino acid, which one is the alpha carbon of the second amino acid, which one has a high energy bond. Where is the peptide bond? Very simple. Peptide bond is always between the carboxylic terminal and the amino terminal. And within that, the carboxylic terminal donates an OH, and the amino terminal of the other alpha, amino terminal of the other amino acid is going to donate a hydrogen. You're going to have an O and two H's, and you're going to remove water, dehydration, synthesis. Now you have a peptide bond that is formed. Now, what does that look like? This long linear bead of this structure of long linear bead of multiple amino acids connected to each other by what? That's aspartic acid, that's glycine, that's phenylalanine, that's glutamate, glutamic acid, this is glutamate, that's glutamic acid, that's alanine, that's arginine, that's aspartate, that's cysteine, that's leucine, that's isoleucine, tryptophan, proline, tyrosine, serine, methionine, lysine, valine, histidine. So they're all connected like that with a peptide bond. This protein structure is what did Christian Anfinsen discover when performing this? Can somebody articulate for us nice and loudly for the whole class what led to the Nobel Prize in the Christian Anfinsen's experiment? Please raise your hand. Somebody raise your hand. Yes, please, in the back. Your name is? In the very back with a hat. Matthew? Okay, Matthew, please go ahead. The primary structure is the most important. Who can add more details to that? Like what is more important about it? Yes, sir. Your name is? Aiden.
When the pH and the heat are set back to the regular setting, the structure like pulls back up into their normal structure. So tertiary, quaternary.
So the normal primary structure has the transcript for the final, terminal, quaternary, tertiary, or secondary, depending on what protein you are. Are you a collagen? Are you keratin? You're a secondary structure. Are you myoglobin? You're tertiary. Are you hemoglobin? You're quaternary. Well, it doesn't matter who you are. Your primary structure is the one who determines that what is is going to be your transcript. Very good. Okay. So the primary structure is a linear sequence. You need to remember that. It's a linear sequence. It is joined together by peptide bond. It's translated from mRNA using the genetic code. We discussed that. And the synthesis begins at the amino end, and we keep adding to the amino end until we terminate at the carboxylic end. Ultimately determines the properties of a protein. So that shows us here, this is the amino terminus. We keep adding to the amino terminal, and we terminate at the that's the carboxylic terminus. Carbonyl group with the O minus. And that's the amino terminus with a nitrogen group. That's the free alpha amino, and that's the free carboxylic group. That's the long linear. So you can tell these are all different amino acids. You see, that's the alpha carbon of amino acid number 1. It has 2 hydrogens. Who is that? It has 2 hydrogens. Which amino acid has 2 hydrogens? Original one and an R substituent who's also a hydrogen. Who is this amino acid? that has an amino terminal and a carboxylic terminal, and not one, but two hydrogens. One is originally terminal, and the other one happens to be the R substituent is also hydrogen. Glycine. And you see this one is phenylalanine. This one is aspartate. That one is threonine. That one is alanine. And you see that's a long chain. Yeah. So you need to know the letters, but it's not that difficult. When you look at the chart, you remember, you know the name, GLY. The other one is glycine. PHE, phenylalanine. ASP, aspartate. THR, threonine. ALA, alanine. ASN, aspartate. So you see now, that's how we make the peptide bond. And then the peptide bond starts to perform a resonance. So because there's steric hindrance, and you see a couple of oxygens that are negatively charged, you know, same charges repel, different charges attract. Okay, you see that shows us you should always be able to tell where do we find the peptide bond. The peptide bond. You see, the peptide bond is very stiff and doesn't allow for rotation. It fixes the, I have no idea who that is, okay? And the peptide bonds are, you see, a very stiff, so they hold the structure of the amino. It's a very tough bond. So it's not like a hydrogen bond. And you show that this is found in the, don't worry about the Ramachandran plot. It just shows you that the energy that is found in each bond. Secondary structure, an alpha helix. You see the dashed lines? What do these dashed lines remind you of? So that shows you that the primary structure had an R group with intermolecular forces that are susceptible to breakage with heat or with acidity. So is secondary structure only alpha helix? So alpha helix is like that. And you see that there's hydrogen bonds to stabilize the structure. And it looks like, and this is the beta pleated sheet. You have a parallel and anti-parallel formation. And you see that there are amino ends and the carboxylic ends of both amino acid peptides are going to be on opposite side. And this is the secondary structure. You see this represents the beta pleated sheet, while this here represents the alpha helix. So the Ramachandran plot shows us that there is a high energy bond that is shown around complexes where, you know, ionizable groups are close to each other because there is constant repelling. They're not going to be attached to each other. Secondary structures. Examples of that would be fibrous proteins like collagen. Remember we said collagen, keratin, collagen. What's the name of the disease? The genetic disease. If the patient has it, they cannot make collagen, and that's why their joints are so wobbly and they cannot, what call that? Polio. Excellent. Polio. I never said the word polio in this class. No. Ehlers Danlos disease. Excellent. Very good. Very good. Very good. Okay, so connective tissue disease. Keratin. What about what is fibrin? Fibrin is the protein that is dropped on the scar tissue. The scar tissue. Scar tissue that is, you know, when you're healing and you're injured a non-regenerative tissue, or you have injured very deeply where the stem cells have been scraped off. So now you cannot enter the cell cycle and perform mitosis no more. So now you will need TGF beta, tumor growth factor beta cells to come and initiate fibrin deposition. This fibrin is secondary structure. Fibrin, tomato, tomato. Okay, yeah. No, it's okay. Yeah, you're good. That's good. That's good intuition, but it's not. Okay. All righty, so if you look at hydroxyproline, what should you expect? If I show you an image of a protein that has hydroxyproline on it, you should immediately deduce what? That no, proline is a ring between the R group and the amino group. We talked about that in the last PowerPoint. That you have the R group and the amino group, and both of them are attached with a ring. So this is a very absurd structure because it causes like a twisting in the structure of the amino acid. You see, that's the hydroxyproline. So not only does collagen have proline, it has hydroxyproline. So that means the proline has a post-translational modification on it. What's the post-translational modification? Hydroxyproline. You see, that's a proline helix, and you see that it is rich. You see, collagen is rich in glycine. So this is just introducing you to what collagen is made up of. It has a proline, hydroxyproline. It has proline in helixes. It has a lot of glycine on it and has some lysine groups. That's keratin. It's found in hair and it's found in the nails. They should have put a picture of a hair of a nail because we want to learn about our own body. Now they're going to put a picture to show you that it's also found in some other animals. This is what is this? This is a deer. Hmm? What is that, a deer? Hmm? Grizzly? A what? Caribou. 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 Never heard of that. Caribou, okay. Sounds interesting. Is it delicious? It's not? I never ate it. I'm thinking. Seen hair, nails, horns. Fibroin can be found in silk, can also be part of the skin. So I have alpha helixes. These are beta pleated sheets. Okay, we talked about that. So that shows you how some amino acids are hydrophobic, some amino acids are hydrophilic. And you know that if some amino acid is soluble, you know that some proteins are membrane bound. We discussed that. We said that proteins are not only enzymes, they are receptors, they are signal transduction, cascade molecules, they are enzymes, they are transcription factors, they are carriers like albumin in the blood. So some proteins, their job require them to be amphipathic. Amphipathic meaning 2 in 1. So one side of that protein is acidic, another side is basic, one side is hydrophobic, another side is hydrophilic. What it needs for its job. If it needs to be, like for example, this ion channel is a protein. That means it's a channel on the plasma membrane. And you know the plasma membrane is bilayer. It is amphipathic. It has hydrophobic tails and hydrophilic heads. So if you're going to squeeze in a protein in the middle, that protein needs to have a polar head and a nonpolar core in order to be embedded in this plasma membrane. Those are the alpha helices. Those are the beta sheets. They combine together to give you the tertiary structure. So now we fold this globular protein, and that is, you see, if it's unfolded, it's like a straight, long primary structure. When the hydrogen bonds start to come in effect, then it's going to fold into a secondary, tertiary structure. What does he do to a folded protein? Our friend said defolded. I agree. But what do we call that? Denature. Very good. Hydrogen bonds stabilizing tertiary structure. You see? And disulfide bonds, I told you about that, called disulfide bridges. Disulfide bonds or disulfide bonds or disulfide bridges. And you see here, you see now that's a force that's going to stabilize the tertiary structure. What are the forces stabilizing a tertiary structure? Hydrogen bonds, that's one force. Disulfide bridges, a second force. Hydrophobic interactions. You know, some amino acids are nonpolar, so they hug each other in the core. So that's a force also. So nonpolar molecules, they are afraid of the polar aqueous solution when you drop the amino acid in water, but they cling on and they hold each other. And you have metallic bonds. Sometimes you have like some metal ions. They break forces, stabilize. So denaturing or unfolding or defolding means that we break the forces, stabilizing the protein. You need to know that. What's the name of a solution that Christian Anfinson would have used to denature and break apart the disulfide bond? You need to remember, are disulfide bonds covalent or are they ionic bonds? Covalent. They are covalent. So that means they are very, very tough. Yes. Did you ask a question? Yeah, we're good. So the disulfide bond is covalent. So you need to use mercaptoethanol. You need to remember that. Who is mercaptoethanol? Mercaptoethanol is a chemical agent that we use in the lab if we want to break apart disulfide bridges. That means there's going to be sulfur in there somewhere in order to break that bond. Detergent. The detergent is a type of substance that is going to destroy and dissolve all hydrophobic substance. For example, we say that salt dissolve in water and nonpolar substance dissolve in nonpolar substances dissolve where? Nonpolar substances dissolve in detergent, in oil or detergent. So that's why they say the stuff that we need to use to wash your, because you know dirt, is dirt polar or non-polar? Dirt is like oil, so it's non-polar. All dirt is non-polar, so we use detergent to liquefy and dissolve all of the dirt and remove it. Okay, questions? Yes. Yeah, if we want to break apart a protein that is attached to each other with hydrogen bond, we heat it because heat breaks hydrogen bonds. We put acid breaks hydrogen bond. We disrupt the pH. If it has disulfide bridges, we add mercaptoethanol. If it has a lot of hydrophobic interactions, we put a detergent in there. Detergent is going to break it apart. The heat, pH change. Oh, another agent we use, chelators. Chelators, that's, you know, some ions like here, metallic ions. Sometimes they're metallo substances like iron, like magnesium. We use those in order to break up. We use chelators to break them. So once you add chelators, then you have broken apart any bond that was made through the ion. Any questions? Are there any questions? No. Okay. A leucine zipper is a type of domain in which we structure some particular amino acids in their secondary structure to fit that leucine zipper. Sometimes another domain, a domain is a way in which proteins have been grouped into groups. How are some alpha helices, or in their conformational structure, they make a zipper type of shape called the leucine zipper. Sometimes they don't structure to make like a zipper shape. They make a shape known as a helix turn helix. And an example of that would be proteins that are like polymerases that transcribe the DNA. Sometimes some proteins are found in structures known as zinc fingers, where there's a lot of metallo ions that hold together the structures of these particular ions. Sulfur domains, Blextrin homology domains. These are found in the signaling type of proteins. Okay, you need to know something very important. Anybody heard of mad cow disease? Yeah. Mad cow disease is when you know your body is equipped with built-in systems that are able to recycle misfolded proteins. You know, these proteins are going to fold into a secondary structure, a tertiary structure, or quaternary structure. This folding process is not random, it's not haphazard. This folding process is highly, tightly regulated. Sometimes due to a mutation, or sometimes when we get really old and the body has been working for so long, sometimes when people, they drink the alcohol or do drugs like cocaine, they interfere with both systems, the systems of folding the proteins, and there are recycling systems that clean any misfolded proteins. So when your body makes misfolded proteins, damaged proteins, there are recycling police systems that come and do housekeeping and take all of the misfolded proteins, crush them, chop them up, and remove them. But what happens when misfolded proteins start to accumulate and pile up and pile up and pile up? They're going to cause damage in the brain. That's what mad cow disease does. Mad cow disease is when the cow has accumulated so many misfolded proteins on its brain that its system of recycling, there's so much turnover, we cannot remove these misfolded proteins that cause brain damage, the cow dies. People do that too. It's called Creutzfeldt disease. And when humans have it, we don't call that mad cow disease. We call that Creutzfeldt disease. Creutzfeldt disease is when the people snort cocaine, when people do substance abuse and drug dependence. Drugs will always increase the rate of misfolded proteins in the brain, and they will destroy the recycling system. So anybody who does drugs, the misfolded proteins are piling up and piling up and piling up until one day they end up with Creutzfeldt disease, and they have brain damage, and they have like exactly mad cow disease. At least the cow did not do drugs. Okay? Especially cocaine and heroin. Heroin and heroin. Okay? Yeah, don't do heroin. One of my students said, don't do heroin. You see, those are the proteins. Sometimes when your body makes a folding error, the police system is built in to take any misfold. We scan. Your own body is always scanning any misfolded protein, any improperly folded protein, any protein that has not been folded properly, and that kind of thing. But when you do that type of drugs and the alcohol and all of the like the unhealthy things, they start to pile up in the brain. And the soda. Soda is bad. Yeah. I stopped a lot. I improved a lot. Oh, prion. What's a prion? You know, the virus used to celebrate that it is the smallest infectious disease. The prion said, no, I am, because a prion is the misfolded protein. That misfolded protein is like a pickpocketing. It's like, because it's misfolded, is not packaged properly, so it looks like a knife. So now it's going to damage the brain, it's going to damage the body. It's called a prion. When a protein is misfolded, it's going to be called a prion. Amyloid disease is a collection of improperly folded protein aggregates in the human body. When misfolded, they're insoluble and they contribute to 20 human diseases, including 4 neurological defects. Alzheimer's disease, that's a buildup of so many years. So as you get really old, I mean, you're going to get so old at some point and you're going to say, turn on the light, and they're going to tell you there's no more light. We can turn more than this. This is the most light we turn for you. Okay, so it's not an issue with the light, it's the issue with your cortex. See, because there's so much accumulation of the improperly folded protein. It's just part of life. That's Alzheimer's disease. Parkinson's disease is a lot with damage, especially people that, you know, like the boxers and people who get a lot of, you know, trauma to the head. Huntington's disease is also a disease where there's an accumulation of misfolded proteins. Rheumatoid arthritis is when there is misfolded proteins that accumulate and your body assumes that they are some foreigner antigens and start to destroy your own body. Dementia is when there is a degenerative, is like when there's a sequela of, as you get older, there is a neurodegeneration. Your cells are not as viable as they used to. The longevity of the cells were not as they used to. So dementia is not like Alzheimer's. Dementia is when your own brain is losing its capacity, when the quality and the structure and the function deteriorate. It's the age-related deterioration of the brain. Yeah, so the capacity, the performance. Bruce Willis.
He's not that old.
Is that the Batman guy? Is A Good Day to Die Hard? I heard about it. No, there's a movie called It's a Good Day to Die Hard.
Yeah. No, he's not in that movie. Die Hard. No, but
I don't watch movies. I read the book only.
Oh, you never watched.
So what happened to him?
He has really bad dementia, he can't even speak. He's not old.
Yeah, you don't know his lifestyle. You don't know his lifestyle. You know, maybe.
Drugs.
Of course.
But he's a what's it?
You know, these rich people, they have too much money. They don't have to suffer in life, so they go dark places. You know, pleasure for them is not, nothing makes them happy anymore. So they go to very dark places and then they have the diseases.
Professor, I have a question, please.
So sometimes poverty is a blessing. Yeah. Yes.
Is dementia like hereditary or?
No. Dementia is based on your lifestyle. Dementia is based on your lifestyle. One of my students said that she works in a nursing home and guys, told me that some people are working in a nursing home with dementia patients. Some of them were professors because they study a lot. What are you talking about? That's a good thing. It causes neuroplasticity. But dementia is for people that maybe overexhausted their brain or never used their brain, or maybe like like their lifestyle, alcoholism, that kind of thing is very effective.
So it's not hereditary.
It's not hereditary. Not hereditary. It's your life choices. Okay. Also living in urban industrialized societies. You know, that's also could affect dementia. Okay. I have no idea who that is. Okay. I know former student, but who is it? I have no idea. Okay. Proper folding. Environment. Okay, so we need to, so we're gonna learn that the proteasome is a collective catalog of all the proteins that we're going to be making. In so what is ubiquitin? Ubiquitin, that's what I was saying earlier. If any protein is improperly folded, misfolded, we're gonna tag it with ubiquitin. How do the recycling systems know that this protein needs to be recycled and chopped up and destroyed? Because there's a tag on it, ubiquitin tag. If you are a protein, you hope there's no ubiquitin tag on you, because if there is a ubiquitin tag, it's a very terrible thing. They cheat me as always. A little better. Very good. Very bad tripod. Okay. All righty. Proteasome. What's a proteasome? Proteasome is the shredder of proteins. Proteasome is the shredder of proteins. Shreds all the proteins. Intrinsically disordered, we talked about those. Primary, secondary, okay, we're done with that. Lastly, we're going to end with hemoglobin. Hemoglobin. Okay. The activity of hemoglobin is based on the metabolic demand. Are you running the marathon? Are you exercising? Do you need oxygen? Then absolutely your hemoglobin is going to be operating at maximum capacity. So animals have widely varying needs for oxygen, and the demand can change. And it operates, so we cannot rely on diffusion like oxygen. We cannot rely on oxygen diffusing from the lung into the blood, and then traveling in the blood and diffusing from the blood into the tissue. That doesn't make sense. So we need an actual transporter of oxygen and carbon dioxide. That's not just going to be diffusion. It's going to be hemoglobin. It's more efficient because we need it for exercise. We need it for survival. If you're being chased by a bulldog, fight or flight, the ATP energy is produced aerobically 15 times more efficiently than anaerobically. And, depends on the availability of oxygen. If your muscle tissues have a lot of oxygen, they will go through the aerobic respiration. If there is no oxygen, how are you going to do aerobic respiration? You're going to do anaerobic respiration. You're going to do an alternative pathway. The classical pathway is to make ATP through aerobic respiration, oxidative phosphorylation. Well, there is no oxygen. You're running on the treadmill and you're consuming all the oxygen. So now you're going to take the other alternative pathway, which is the anaerobic respiration or fermentation. is an efficient and adaptable oxygen delivery is necessary. You need to know this. This is one of the questions on the exam. The hemoglobin is a coordinating structure, is like 4 proteins in one. And it's actually really tiny. Hemoglobin is enormously tiny. An oxymoron, enormously tiny. Hemoglobin is enormously tiny. And it has 4 subunits. You need to know 2 alpha and 2 beta. Which hemoglobin has 2 alpha and 2 gamma? The baby, the fetal hemoglobin. That's the regular hemoglobin. You need to know the hemoglobin, each subunit has 1 heme group on it, and each heme group on it has double dioxygen. You need to know that. So you see, it's like a donut hole. It has like a hole in the middle in order to trap the oxygen. It has 4 subunits. Each subunit has 1 heme group, and each heme group is going to hold 1 dioxygen, not 1 single oxygen, 1 dioxygen. Myoglobin is, this is, students think that myoglobin is fetal hemoglobin. No, myoglobin is tertiary structure. Myoglobin is the muscle hemoglobin. It has 1 subunit, not 4, and it has 1 heme, and each heme is going to hold how many oxygens? Dioxygen, O2. Okay, you need to know that heme has a prosthetic group. You see this metal prosthetic group, and it's a ferrous, not ferric. Ferrous, Ferrous. What did you need? Yeah, what is it? What is going on? Okay, so this iron is a metal, and you know you have 4 groups, 2 alpha and 2 beta. The 2 alpha are purple and 2 beta are red. And on each group, whether alpha or beta, you have 1 heme, and 1 heme is gonna bind oxygen, dioxygen, O2, and the heme is in the ferrous state. So you see that ferrous, if you are in the ferrous iron, the methemoglobin is not going to work. Only in Fe 2 pluses is going to bind oxygen. Ferrous is 2 pluses. You see, this is the structure of hemoglobin. And you know that histidine is an amino acid that is part of your hemoglobin. And when there is no oxygen, deoxyhemoglobin, meaning that hemoglobin doesn't have an oxygen, when you add oxygen to this empty deoxyhemoglobin, now you're going to have oxygenated hemoglobin. The histidine is going to move out of the way so the oxygen can bind into the ferrous state. So remember the cooperativity we talked about earlier. Look at this. This is a hemoglobin that is empty. Each circle means oxygen bound. So oxygen number 1 bound. Then number 2 came along. Then number 3 came. Then number 4. So now we have fully saturated hemoglobin with oxygen. And now we have exited the lungs. Questions? That's the cooperative binding. Entering the lung, you need to know. Entering the lungs, you need to know the difference between a T state and an R state. What's the difference between T state and an R state? R state means that hemoglobin is hugging all the oxygens and never released a single one. The T state means it released all of them and it has nothing in the back. R state, I'm full of oxygen, haven't let go of a single one. T state is when we let go of the oxygen. You're gonna learn that smoking, people who smoke nicotine or people who smoke the cannabinoids, they are basically suffocating themselves because the nicotine and the carbon monoxide, what it does is it locks hemoglobin in the R state. So it has a lot of oxygen, but it's not letting it go. So now these people who smoke nicotine, how is the bone marrow going to react? You remember this from anatomy and physiology. If the oxygen saturation in your blood goes down and you're suffering from hypoxemia, your red bone marrow is going to make more red blood cells. But it's going to assume we don't have enough red blood cells. But we do. And they are full of oxygen in the R state. They can now release it to the T state and empty it out to the tissue because of nicotine. Now the body makes more red blood cell, more red blood cell, more red blood cell. Now the blood of these smoker people is gonna become thick like honey. It's gonna clot. That's why these younger juvenile, 16 years old, 15 years old, they go to the pediatric, and when they do blood work for them, they have hypercoagulability. Their blood is so thick and their red blood cell count is so high. Why? Because they're smoking a cigarette in a toilet.
What?
And they smoke sumo or wherever they do it.
What about the hookah?
And that too. So whatever that they do, they are, and the mom, oh, not my daughter. Oh, not my son. My son would never smoke. My son would never drink. He's smoking and his blood is so hypercoagulable. But I'm tired. Okay, he doesn't do it. Okay, go. He's smoking. What is the reason why a 15-year-old and a 16-year-old and a 17-year-old, 13-year-old having a hypercoagulable blood, and they never had something like that few years ago because the peer pressure. Because the peer pressure in school, oh, you don't smoke, so now they just gonna follow along. So it's very important where you plant your kids, where you take your kids.
The plants, if they find all the people they still the vape.
Yeah, it's all bad.
And the hookah.
It's all bad. The vape, the hookah, the nicotine, they are suffocating themselves and they're gonna get a blood clot. Because the bone marrow has no way of knowing that. The bone marrow has no way of knowing that we have red blood cells enough, we have oxygen, it just cannot be let out because they are smoking the nicotine. They need to stop the nicotine. So it's going to assume that, hey, maybe because we don't have enough red blood cells. So it's going to be making more and more until they have, a lot of them die with like a pulmonary embolism and blood clots. Okay, you need to know T and R state. T state, we unloaded everything, we're empty, we need to go back to the lung. R state, we are just loaded everything full of oxygen. What does nicotine and all of the smoke lock hemoglobin in? T state or R state? R state. So it has a lot of oxygen, it's just not coming out. So the binding of the first oxygen favors, cooperates, positive feedback. The second, cooperativity. Cooperativity is important as hemoglobin rapidly passes through the lungs. Who is that? This shows you your sigmoidal curve. You need to remember that hemoglobin has an S-shaped sigmoidal curve. You see, when the partial pressure is 25, you see that the oxygen saturation of hemoglobin is also 25. Myoglobin is not like hemoglobin. Why? Because myoglobin is not a transporter of oxygen, it's the storage. One is for transporting. Hemoglobin cannot wait to give you the oxygen. Myoglobin is not about giving you the oxygen. Myoglobin is about retaining the oxygen, holding it for storage. Myoglobin is a storage molecule. Hemoglobin is an exchange molecule. Hemoglobin want to give you the oxygen. Myoglobin will never give it to you. It will hold on to it to save it. for later when you need it. You need to know this graph. One has a sigmoidal shape, the green one, that's hemoglobin, and the other one does not have a sigmoidal shape, the myoglobin, and this shows you that it's storage. So at low oxygen, myoglobin holds more than hemoglobin. That shows you myoglobin is a storage because it holds more. At high oxygen, both hold 100%. That's a sigmoidal binding. So that shows you. Everybody look, myoglobin is better for, is everybody see where my mouse is? Myoglobin is better for storing oxygen, while hemoglobin is better for delivering oxygen. You're going to see that on the exam. Myoglobin is better for storing oxygen. Hemoglobin is better for delivering oxygen. Now, who's that? The Bohr's effect, our good friend. That shows us here. So now look, at pink, pH 7.6. Orange, pH 7.4. Green, pH 7.2. Blue, pH 7.0. Obviously, the more you move to the right, the more stressful or least stressful. What is more stressful? 7.6 or 7.0? Who's more acidic? 7, because acidosis. Is acidosis going to be with 7.6 or 7.0? So acidosis, 7.0. So when you look at acidosis, the curve for acidosis is the one that shifts all the way to the blue. The original one is this one, is pink. The more stressful things, the more we're going to shift to the right or to the left. We're going to shift to the right. That's your right. It's going to shift. It's the rightward shift. That's the graph. Look, that's the graph. And we're going to shift to the right of the, that's the right of the graph. That's the left of the graph. So that means the more stressful conditions get, the more we're going to shift to the right, indicating we're accommodating. We're accommodating. So if you pick any random point, look at this point here. If the pH is 7.6 and the partial pressure of oxygen is 20, you will see that at pH 7.6, you will see that the oxygen saturation hemoglobin is 0.2. If the pH is 7. If the partial pressure is 20, you will see that at 20, now you move up here. At 20, you see that the partial pressure is 0.6. And the more you shift to the right, the more stressful it is. You see that at this point, you see that we unload, that oxygen is going to leave hemoglobin when the tissue is in deficiency of this oxygen. You see more oxygen required to have the same fraction bound. Okay. We talked about that. Oxygen, tissue long oxygen. So acid is going to favor release of oxygen. So if you go to the tissue and the tissue has carbon dioxide, hemoglobin will unload. These are factors, remember, stressful factors induce hemoglobin to unload oxygen, like heat, acid, carbon dioxide. What is this 2,3-BPG, bisphosphoglycerate? That's the metabolic waste, just like carbon dioxide. It means that the tissue is generating so much waste, we need oxygen. So these are all factors. BPG is 2,3-biphospho or bisphosphoglycerate. The more we have of 2,3-BPG, the more oxygen hemoglobin is going to give. The more hot it is, the more oxygen hemoglobin will give. The more acidic it is, the more oxygen it will give. The more stressful things are, the more oxygen is going to be unloaded from hemoglobin. So 2,3-bisphosphoglycerate is a product of glycolysis, experiencing muscle cell rapid use of glycolysis, and it gonna bind in the donut hole. So this 2,3-BPG carbon dioxide, they gonna bind hemoglobin, and oxygen is gonna leave, which was what we need. If you come to a tissue that has a lot of partial pressure of carbon dioxide, a lot of partial pressure of 2,3-BPG, so you want that. You want carbon dioxide to come out the tissue. You want 2,3-BPG to leave the tissue and bind hemoglobin, and you want oxygen to leave hemoglobin and bind the tissue for aerobic respiration and oxidative phosphorylation. Are we clear? Okay. Again, with the Bohr's graph, it shows you as pH moves down, 2,3-BPG goes up, meaning those are those are all stressful conditions. pH goes down, acidosis. 2,3-BPG go up, it means that we have metabolic waste. Carbon dioxide goes up, it means that it's also a form of toxic metabolic waste. So the graph is going to shift to the right to accommodate and release more oxygen. Very good. So rapidly metabolizing cells produce acid, which is going to mean hemoglobin is going to unload oxygen like crazy. Rapidly metabolizing cells release carbon dioxide. It's going to release oxygen. Rapidly metabolizing cells release 2,3-BPG. It's going to release oxygen. All favors oxygen release from the hemoglobin. So rapidly metabolizing cells get more oxygen. 2,3-BPG is a big concern for smokers. So the blood of a smoker has high levels of nicotine, high levels of 2,3-BPG also, because it's a metabolic waste. So the hemoglobin is going to get locked into the T state in the passage through the lungs. Yeah, if you stop smoking. If you stop smoking, it will return back to normal because it is the nitric oxide, it is the carbon dioxide and the carbon monoxide. They lock, they are binding and locking the hemoglobin into the T state.
But if you stop smoking in a learned terms, you know, like your
Yeah.
lungs is damaged.
The hemoglobin is going to get locked in the lung, is going to get locked into the T state, so it cannot uptake oxygen. But if it already has oxygen, once it gets to the tissue, it cannot unload. It does both. So it prevents unload of new oxygen in the lung by locking hemoglobin in the lung in the T state. But in the tissue, it locks it in the R state. Yeah, it depends on the location. Very good. Carbon monoxide also does that. So carbon monoxide binds to hemoglobin a lot stronger than oxygen. So that's why carbon monoxide poisoning kills people, because instead of your body transporting oxygen, it's going to be transporting carbon monoxide, because carbon monoxide is much stronger binding to hemoglobin than the oxygen. Okay. Fetal hemoglobin. Fetal hemoglobin, this graph shows you what? Fetal hemoglobin is stronger in binding to oxygen than adult hemoglobin. Meaning that if a mom is pregnant, her fetal hemoglobin, if one mom adult hemoglobin has oxygen pass nearby, what is the fetal hemoglobin going to do to it? Steal all the oxygen from it. Because the fetal hemoglobin is stronger in binding to oxygen than the adult hemoglobin. So the baby is going to literally, the fetal hemoglobin of the baby is going to be stealing the oxygen from the adult hemoglobin of the mom. You're not going to be nice to your mother. Fetal hemoglobin has 2 alpha and 2 gamma, not 2 beta. 2 alpha, 2 beta for the adult. For the baby, it's 2 alpha and 2 gamma. Talk about sickle cell anemia, and you need to know that It's a mutation from glutamic acid. That's what we want. To valine at which position? Position number 6. And the red blood cells are going to become sickle. They're going to look like a sickle. You know this shape. You know the sickle that they used to use in the farm. This thing here. That like somebody hold it like this, and they have a face like that. Like the person holding it, and then they cut the grass with it. So your blood cell is supposed to look like that. Biconcave, meaning concave from the top and the bottom, depressed from the top and the bottom. It's supposed to look biconcave and discoid and biconcave. But this one is going to become sickle. Why is it becoming sickle bad compared to biconcave? Biconcave means that you maneuver so smoothly, and it's thermodynamically favorable for structure. This one is how your red blood cell is going to move so freely. But sickle cell is not going to move freely, and it's not going to move freely, and it's not going to be able to transport oxygen. So there will always be a metabolic defect. One advantage of having sickle cell anemia is that you will never get malaria. And remember, the sickle shape happens when you are under stress. If two people, one with sickle cell, one is not, if both of them are sitting together, you take a random blood sample and everything is okay, you will never, ever know which one has sickle cell anemia. Only when you ask them to exercise or when they're under stress, then they start to become sickle. Okay? You see, red blood cell, we talked about that. They move nicely through the red blood cells, but sickle cells, they clump up and they make like a clot. They get stuck, and the sickle cells are removed by the spleen. The spleen is the graveyard of the red blood cells. Removes all of them. Benefit of sickle cell mutation for the heterozygous. No benefit for homozygous. So for example, if you have sickle cell anemia, that means you have 2 copies. You have actually sickle cell anemia. That's not an advantage. The advantage if you're heterozygous, meaning that, so if you want to make a graph, if you want to make one square, let's say the father has big R, little r, the mother big R, little r. If you want to make a Punnett square, what's going on? You see that you can either have 2 big R's, big R, small r, and big R, small r, or 2 little r's. This one is the one with sickle cell anemia because it's homozygous recessive. And this one with 2 big R's, it means that you're healthy, you're normal. When you're heterozygous, meaning you have 1 copy healthy, 1 copy disease, that person will never get malaria.
Well, are they carriers or no?
I'm sorry?
The big R and the small R.
They get it. Yeah.
Are they carriers?
If you no carriers of what?
The sickle.
Yeah, of course. If you are big R, little r, this is the disease. So if you have a copy of this, you're a carrier. So if you are a carrier, you will never get malaria.
Oh, really?
Yeah. If you are a carrier, not if you have sickle cell. You have sickle cell.
If you're a carrier and you have age, you have chances.
Yeah, absolutely. Yeah. Depending on, you have chances, absolutely, but depends on the father. So you see that if you have lupus is not related to sickle cell anemia. Lupus is like a nerve defect, like autoimmune disease affecting the nerves, but not your sickle cell. Everybody listen. Shh. What's going on? So if you look here, you see that if you're affected, 2 little r's, you will have the disease. If you are healthy and you're unaffected, you see that the benefit of sickle cell mutation is for the heterozygous. No benefit for homozygous recessive or dominant. So if you're healthy or if you are affected, no. If you are a carrier, you survive. Look, the benefit of sickle cell mutation is for the heterozygous, meaning you have a big copy and a small copy. But if you are homozygous recessive or dominant, meaning you have 2 little R's or 2 big R's, there's no advantage for it, only for the heterozygous. Okay, so this is the summary. Hemoglobin has 4 subunits in hemes. Myoglobin has 1 of each. Binds to oxygen. R state binds to the oxygen beta. T state releases oxygen. And we talked about that. Anybody has a question? Yeah, you gotta upload. I'm gonna open the portal for you now. This is due. Last week, I don't think this was clear, but in class work is due in the end of the lecture and is only for students who were in class. If you're absent, you are allowed 1 excused absence. If you were absent with that excused absence that I excused you, missing that assignment, that 0 is not going to affect you. But you cannot be absent and you copy your classmates' notes. That's only for people who show up to class. So let me take the attendance, please. Hold on. Do not leave yet.
Can you open the